Assalamu alaikum dear students. Today we are talking about MDCAT test 2025 chemistry MCQs quick revision part 3rd. Dear students, if you need MCQs for MDCAT test preparation, let me know in the comment section. Dear students, अभी तक अगर आपने मेरे YouTube चैनल को सब्सक्राइब नहीं किया तो जल्दी से सब्सक्राइब कर लें, लाइक और शेयर ज़रूर कर लें। शुक्रिया। The melting temperature of DNA is increased by dash. Option A, adenine and thymine content. B, guanine and cytosine content. Option C, sugar content. And option D, phosphate content. So the correct option is B, G and C content, guanine and cytosine content. So explanation is, the stability of DNA double helix is determined by the hydrogen bonds between base pairs. 3 hydrogen bonds connect with G and C, guanine and cytosine, while adenine and thymine connected with 2 hydrogen bonds. Therefore, DNA with higher proportion of GC content. So it requires more energy and higher temperature to denature. So this is very important MCQs. What is the melting temperature of DNA? Option A, solid DNA becomes liquid. B, liquid DNA evaporates. C, DNA changes from double helix into supercoiled DNA. And option D, native double helical DNA is denatured. So the correct option is D, native double helical DNA is denatured. So explanation is, the melting temperature is the key property of DNA. So it's the temperature at which the double helix separates into 2 single strands. This process is called denaturation. Nonsense codons are present on option A, mRNA, B, tRNA, C, rRNA, and option D, none of these. So the correct option is A, mRNA. Nonsense codons, also known as stop codons, UAA, UAG, UGA, are specific 3 nucleotide sequence found on mRNA that signal the denaturation, termination of translation. So they do not code for any amino acid, but instead cause the ribosome to release the newly synthesized polypeptide chain. Codons are present on option A, coding strand of DNA, B, mRNA, C, tRNA, and option D, rRNA. So the correct option is C, tRNA. Explanation is, transfer RNA molecules have 3 nucleotide sequence called an anticodon. This anticodon is complementary to the specific codon on the messenger RNA and ensures that the correct amino acid is delivered to the ribosome during protein synthesis. Dear students, subscribe now and press the bell icon. Never miss an update from a technologist. Mammalian RNA polymerase 1 synthesize. Option A, mRNA, B, rRNA, C, tRNA, and option D, hnRNA. So the correct option is B, rRNA. Eukaryotic cell have 3 main types of RNA polymerase, each with specific function. So RNA polymerase 1 is in the nucleus and is responsible for transcribing the genes for ribosomal RNA, rRNA, except for 5S rRNA. Mammalian RNA polymerase 3 synthesizes. Option A, rRNA, B, mRNA, C, tRNA, and option D, hnRNA. So the correct option is C, tRNA. RNA polymerase III transcribe genes for transfer RNA, tRNA. 5 is rRNA and other small regulatory RNA. These molecules are relatively small and have specific, well-defined functions in the cell. Dear students, this is very important MCQs. Mammalian DNA polymerase is in option A, nucleus, B, nucleolus, C, mitochondria, and option D, cytosol. So the correct option is C, mitochondria. Explanation is DNA polymerase is a specific enzyme responsible for replicating and repairing mitochondrial DNA. So its crucial role for the maintenance of the mitochondrial genome, which is separate from the nuclear genome. The other DNA polymerases are primarily found in the nucleus. Non-coding sequences are present in the gene of option A, bacteria, B, viruses, C, eukaryotes, and option D, all of these. So the correct option is C, eukaryotes. Eukaryotic genes are typically composed of coding regions, also known as exons, and non-coding region, introns. The introns are removed during RNA splicing. So this is distinguishing feature of eukaryotic gene structure, as prokaryotic genes are generally continuous coding sequences. The synthesis of RNA and a DNA template is known as option A, replication, B, translation, C, transcription, and option D, mutation. So the correct option is C, transcription. Transcription is the first step of gene expression, whereas a segment of DNA is copied into RNA by the enzyme RNA polymerase. This process is distinct from replication, which is the copying of DNA to make more DNA. The translation, which is synthesis of protein from mRNA template. Introns are present in DNA of dash. Option A, viruses, B, bacteria, C, human, and option D, all of these. So the correct option is C, human. Introns are non-coding segments of a gene that removed during RNA processing. They are characteristic of eukaryotic genes, which include humans, but are generally absent in the DNA of most prokaryotes like bacteria and many viruses. Dear students, this is very important MCQs. Some DNA is present in the mitochondria of option A, prokaryotes, B, eukaryotes, C, both A and B, and option D, none of these. So the correct option is B, eukaryotes. Eukaryotic cell, unlike prokaryotes, contain organelles like mitochondria and chloroplast. These organelles have their own small circular DNA molecule known as mitochondrial DNA, which encode some of proteins needed for their functions. In RNA, the complementary base of adenine is option A, cytosine, B, guanine, C, thymine, and option D, uracil. So the correct option is D, uracil. The nucleic acid's specific base pair occurs in DNA adenine pair with thymine. However, in RNA, thymine is replaced by uracil. So adenine pair with uracil. Mutations can be caused by option A, ultraviolet radiation, B. Ionizing radiation, C. Alkylating agent, and option D. All of these. So the correct option is D. All of these. Mutagens are agents that cause mutation. UV radiation, ionizing radiation, and alkylating agents are all well known as type of mutagens. UV radiation causes thymine damage. Ionizing radiation creates free radicals that damage DNA, and alkylating agents add alkyl groups to bases, altering their pairing properties. Substitution of an adenine base by guanine and DNA is known as option A. Transposition, B. Transition, C. Transversion, and option D. Frameshift mutation. So the correct option is B. Transition. A transition mutation involves the substitution of purine base adenine and guanine, adenine or guanine, for another purine or pyrimidine base, cytosine or thymine. For another pyrimidine, since adenine and guanine are both purines, this is transition. A point mutation results from dash. Option A. Substitution of a base, B. Insertion of base, C. Deletion of a base, and option D. All of these. So the correct option is D. All of these. A point mutation is a change in a single nucleotide base. This can occur through the substitution of one base for another, or the insertion or deletion of a single base pair. All these single base changes are considered point mutations. Dear students, this is very important MCQs. Substitution of a base can result in a dash. Option A. Silent mutation, B. missense mutation, C. nonsense mutation, and option D. All of these. So the correct option is D. All of these. A silent mutation occurs when a new codon codes for the same amino acids. A missense mutation occurs when a new codon codes for different amino acids. A nonsense mutation occurs when new codon is a stop codon, resulting in the premature termination of the protein. A silent mutation is most likely to result from a dash. Option A. Substitution of the first base of a codon, B. Substitution of third base of a codon, C. Conversion of a nonsense codon into sense codon, and option D. Conversion of a sense codon into nonsense codon. So the correct option is B. Substitution of third base of a codon. The genetic code is degenerate, meaning that the most amino acids are encoded by more than one codon. Often, the first two bases of codon are most critical, and changes in the third base, the uval position, are less likely to alter the amino acid that is specified, leading to the silent mutation. Dear students, if you like this video, please सब्सक्राइब माय यूट्यूब चैनल। अगर आपको ये वीडियो पसंद है, बराए मेहरबानी मेरे यूट्यूब चैनल को सब्सक्राइब, लाइक और शेयर ज़रूर कर लें। शुक्रिया। DNA dependent RNA polymerase is option A monomer, B dimer, C trimer, and option D tetramer. So the correct option is D tetramer. Dear students, this is last MCQs. Hemoglobin S is an example of dash. Option A silent mutation, B acceptable missense mutation, C. Unacceptable missense mutation, and option D. Partially acceptable missense mutation. So the correct option is C. Unacceptable missense mutation. Explanation is hemoglobin S is the cause of sickle cell anemia. It results from a single base substitution that changes a glutamic acid into a valine. This change is considered unacceptable because it significantly alters the structure and function of hemoglobin protein, leading to the severe disease phenotype. Thank you for watching my videos.